I'm going to make it fun. Okay. So let's get ahead. Where you start working. There we go. Okay. Let's flip on through and okay, it's recording. All right. I like talking about inflammation because we get to start to talk about some of the cool aspects and the steps of what the cells do when they're leaving the bloodstream and moving out to the tissue. And it's actually really kind of interesting to learn about. So let's first describe or define what inflammation is: an immediate response to ward off unwanted substances. So two descriptors that are up there on the board tell you, one, that it's local, which is good because you don't want inflammation occurring throughout the entire body, right? You sprain your ankle, you don't want your hands swelling up. You know what I mean? So that makes sense. And it's non-specific. But here's the thing, and I can't remember if I've asked this on a test before, but I'm almost positive I have. Inflammation is associated with vascularized tissue. Okay, vascularized tissue. So you have to have blood vessels. So you're not really going to see inflammation in avascular tissue, for example, the epidermis. Right? Inflammation will usually be underneath that in the connective tissue or in the dermis, but it won't be in the actual epidermis per se. All right, it has to be vascularized tissue. All right, I'm not saying it's not going to happen, but it's very unlikely. All right, so innate immunity. So let's talk about what happens. So this is a very simplistic way to describe how inflammation occurs, but it usually starts off with some tissue, something gets hurt. So you start off with inflamed or injured tissue, and then we're going to have our cells that are going to play a role in that. And we already know, I feel pretty confident, I hope you feel pretty confident in the roles of what basophils and mast cells do. All right, so we're going to involve those two cells. And then, of course, if we have injured tissue, like that example that the book uses, the person skinning their knee, we're going to have some infectious organisms that get in there. So something happens to the tissue. These cells are going to play a role in that, and infectious organisms are most likely going to be involved because they're present. So the injured tissue and these infectious organisms and some of the other cells are going to release chemicals that are going to cause inflammation there. So all of these things listed here play a role in that. And so, of course, when we talk about the chemicals that are going to be released, we know that the basophils and the mast cells are going to release histamine, and that's going to make our cells leaky or permeable, right? Then we have our leukotrienes and prostaglandins. Those are the eicosanoids that are going to be released by injured tissue. And then our, what we call the chemotactic factors, that's our signal flare. That's going to be, all right, the substances that the cells, the infectious agent, will be releasing to draw more immune cells to the site of injury. All right, so we'll go through the steps. It's kind of a neat kind of process here. All right, so let's start off here. All right, the chemicals are going to cause some changes in your blood vessels there. So the first thing is going to be vasodilation. What's that going to do? What is vasodilation going to cause? Nailed it. More blood flow. Yep. Let's get more blood flow there so we can get more stuff to the area there. And we're also going to see more leakiness. And this is new. We haven't talked about this, but we're going to see these special molecules called CAMs, which stand for cell adhesion molecules. And normally in the endothelium is slippery. We learned about this when we talked about hemostasis, remember? And how those cells are producing, right, the prostacyclines, the prostaglandins, which make it slippery so things don't attach onto the endothelium. Because if you start attaching things onto the endothelium, you can start to get, all right, blockages. I don't want to say blockages, but a buildup, like you can get that atherosclerosis, for example. All right? So we don't want that to happen. Except, okay, we don't want the cell walls to be sticky until this happens. So you get tissue damage, and we're going to have the cells, the endothelial cells, are going to release, or not release, but they're going to create these things called cell adhesion molecules. The best way I can describe that is someone falls into the river and they're getting washed down the river. And you got like 100 people standing on the riverbank. And those 100 people aren't going to get into the river, but they're going to extend their arms out to try to grab onto that person that's flowing down the river. That's what these things do. The cell adhesion molecules do exactly what the name says. They're going to try to grab onto cells, namely white blood cells, so they don't flow on past the site of injury. They're going to stop and slow the cells down so those cells can then migrate out of the bloodstream there. Now, I'll talk about this, right? So keep in mind, these three things happen. Vasodilation increases blood flow. We're going to make the blood vessels more permeable, okay? And then finally, we're going to put these things on the outside of the endothelium. And the endothelium is going to be the simple squamous epithelial cells that line the internal lumen there of our blood vessels. And they're going to make the endothelium more sticky by putting these molecules out to start catching cells. Right? So how does this happen? So these cell adhesion molecules are going to then start to catch these cells. And the first part of this is called margination. So the cells are going to move to the periphery or the margin or the edge of the blood vessel, and they're going to get stuck onto the endothelium by these cams. So once they're stuck on the edge of the blood vessel, now remember, our blood vessels now are leaky. So now you'll have these holes in between the endothelium. And so these cells, when they get stuck on the edge of the endothelium, they'll migrate over towards these holes here and they'll squeeze through. It's really kind of a cool process. And that process is called diapedesis. Cool name. So diapedesis is when those cells on the periphery start to squeeze through those holes, those gaps between the endothelial cells, and they move out into the tissue out here where there's pathogens and whatnot, okay? So the third thing that's going to happen is chemotaxis. Because remember, our white blood cells do not have eyes. They do not have ears. They can't find their way just by themselves. So the third part is that chemotaxis, because the chemicals that were released from the damaged or dead cells are going to be giving off these chemicals, which will attract our white blood cells. And then if any pathogens manage to get into the tissue from that knee scrape, they too are going to give off those chemotactic chemicals, which attract our white blood cells. You kind of see how this process is working, right? We want to get stuff from the blood into the tissue. To do that, we got to make our blood vessels leaky, right? And then we've got to increase blood flow. And then we got to make the endothelial cells near the site of damage sticky so they can slow the cells down that are flowing by, get them to attach onto the edge or the margin of the blood vessel, and then they can squeeze through those gaps that we created with the histamine. They'll squeeze through the gap, and then they're just going to kind of follow their senses towards where those chemicals are in high concentration, because that's where the problem is. And then they're just going to start doing what they do, right? If it's a macrophage, it'll start going after the pathogens, and it'll start to clean up the damaged tissue there, just start gobbling up and eating up the damaged cells. Okay, so it's a cleanup crew. So these white blood cells are going to, at the same time, also release more chemicals. How do you like this? And we're going to make more white blood cells. Because we're going to lose some white blood cells. It's a battle, okay? The immune response is an ongoing war that happens every day. And so you're going to lose some soldiers. So we've got to make sure that the soldiers that we lose, we can replace. So that's what we're going to do. We're going to upregulate leukopoiesis in the bone marrow so we can get more soldiers out there. And then our macrophages, they can release a substance called pyrogens, which is going to stimulate the hypothalamus to increase, right, your body temperature. And we'll talk about, when we get to the fever, why that is. But it's actually a benefit, right? We'll talk about all that goes on. All right, questions so far? Not too bad, right? Not too bad? Okay. All right. Then, remember what we said, and I said this before in our last class, when we make our blood vessels leaky, stuff is going to come out of your blood, right? Fluid mainly, but it's going to wash out your plasma proteins. And so with those plasma proteins, we're going to effectuate, all right, the colloid oncotic pressure there, right? But what else is going to come in our blood plasma? Well, we love these guys. Those are your antibodies, immunoglobulin. We just learned about this stuff, complement. Okay, we're going to see how complement is going to help with the immune response there. And then back in chapter 18, our blood clotting proteins. So we got to clot up the bloodstream so we're not losing blood. And then, of course, we talked about the effect of kinins there, how they affect the pain response and whatnot, and they help promote the clotting effect there. All right, so clotting proteins, just talked about that. We're going to separate out and keep those microbes from getting into our bloodstream. And then our kinins here, pain receptors, but what else are they going to do? They're going to also make your blood vessel leaky, and they're also going to make more CAMs, which makes our endothelium sticky. All right, sticky. All right, thoughts on any of that? It's not terribly exciting, but it's interesting, I would say. So as the proteins leave the blood and migrate out into the tissue, we're going to see a movement of fluid from your blood into the tissue. And so we call that fluid exudate. Right? So exudate is that fluid that comes from the blood out into the area in the tissue, which can be injured or affected there. And so understand something about this here. A lot of things will cause this to occur. And I kind of talked about how all this occurs. All right. And you kind of know it. We just got to, it's like a story. We got to piece this story together here. So that fluid that is coming out of our bloodstream, just to repeat myself, lots of plasma proteins, right? Immune cells that are in our blood. All of this is going to help take out the pathogens, but also it's going to help with the healing process. So that vasodilation increases blood flow, increases all that material coming to the area. And then we are also going to see an increase in that leakiness of the blood vessels there. And I already mentioned this part, when we start to get those plasma proteins into the tissue, right, this is going to effectuate the osmotic pressure. And so we're going to see an increase in fluid buildup initially in the tissue there, right? But eventually, okay, we have the lymphatic system that's going to help drain that off. And then as we start to go through the healing process and we've taken care of the injured tissue, you'll start to see the blood vessels become less leaky. When they're less leaky, we won't be losing all those plasma proteins into the tissue. And so then the osmotic pressure will shift back in favor of keeping fluid back into the blood vessels there. Right? But keep in mind, this is what I said in chapter 21, our lymphatic vessels are going to help take up that extra fluid. And that process is called washing, which is important. And I'll explain all this because as that extra fluid comes in, it kind of washes through the tissue there. So you've got, it's like in your bathtub, you've got a drain that's going to drain everything off. And that's usually what the capillary bed is going to do, except it doesn't get everything. Okay. And then as that process, we get the drain on one end and the fluid coming in at the other, it washes across and goes out through the drain. So that's going to move stuff through the tissue there. And so essentially, it's going to help to carry away a lot of junk. And which is good because here's the thing. When we wash things into the lymphatic system, we just talked about in chapter 21 in lab what the lymphatic system does, right? We talked about lymph nodes. We talked about the spleen and all those secondary lymphatic structures. They're going to be loaded with white blood cells. So all that stuff that got washed out of the tissue is now going to go through your lymphatic system where you have white blood cells evaluating and analyzing all of that stuff that just got washed out of that tissue site injury there. And so there's probably some pathogens there. So we can kind of take care of that stuff. So this is the amazing thing. Inflammation, 72 hours. I'll even go as high as 96 hours. All right. Good thing. We want that. Okay. Chronic inflammation could be a bad thing, and I'll address that later on. Right? But after 72 hours, things start to slow down. Pretty much, hopefully, we've stopped any bleeding that's going on, right? But also, right, we've handled what's going on in that tissue locally. And now we've just got to get rid of, the fallen soldiers, so to speak, and start repairing the damaged tissue. That's when the fibroblasts come in and they start laying down collagen to help the healing process there. All right, so this here, I love this picture. I'll go through it, kind of show you in picture form. All right, so if you look here on the left side of the screen, so big picture, all right, this young person here was skateboarding, they fell off, and then they scraped their knee. So they've compromised the cutaneous membrane. Their innate immunity has been compromised now, right? The barrier has been breached. You've got pathogens that have managed to make it into, all right, deep into the dermis there or the connective tissue below. So we have all this stuff going on. So you've got damaged tissue is going to be giving off chemotactic chemicals, right? And these bacteria here are also going to be giving off chemotactic chemicals. So the signal flares are being shot up. Now, if there's any white blood cells here in the tissue, they can immediately go to work, like dendritic cells, macrophages, whatnot, can start to go to work here. So now let's talk about what happens, all right, during inflammation. So step one here, you get those chemotactic chemicals that are being released here, but also the mast cells that are in close proximity to the small blood vessels, they're going to start to release histamine and heparin. So we're going to create permeability here in between the endothelial cells here. And then also we're going to prevent it from clotting. So you got mast cells on the outside, basophils on the inside. They're working towards the same goal there. So they're going to be releasing those chemicals. Whoops, gone too far. And so that's going to cause, all right, our separation here, making it more permeable. Now we're going to see vasodilation of the blood vessel to increase blood flow. And then finally, we're going to create those CAMs, those cell adhesion molecules, those little sticky things, and they're going to start to try to grab onto these cells as they're tumbling and rolling through. So we're going to grab onto them, and we're going to try to get them to come out of the blood vessel. All right, so that's what we see here in step 3, right, when we talk about the recruitment part. So we're going to get margination, so this is going to start to hang out close to the edge here of the blood vessel. And then we're going to get diapedesis, where it starts to squeeze through. And then finally, the injured site here is giving off all of those chemotactic chemicals. So then the white blood cell, this neutrophil here, will start to migrate through, right, the tissue and get over to where the pathogens are. And so it'll start to gobble things up. Now, meanwhile, we still have that leaky tissue. So what else is leaving? Right? Plasma proteins are leaving. So guess what? That's going to cause water to also, or fluid, I should say, to also leave, right, your blood vessel. So antibodies are leaving, right? Clotting agents are leaving, which is what we want, because we want to clot things up, kind of block things off. So this process here, as we start to increase the fluid content here in the tissue, on the far end of our picture here, you got your lymphatic capillary there. And so the lymphatic capillary is going to be draining stuff from the tissue into your lymphatic system. Remember, I talked about pressure gradients. So we're going to start to build up the hydrostatic pressure gradient out in the tissue while it's lower in the capillary. So that's going to promote fluid to move into the capillary. As it does it, it just kind of washes things away. So it's going to wash away cell particles. All right. Whoops.
If there's like too much fluid, is that when you get like serious drainage?
When you say,
So like when like the proteins are coming out and it's like drawing the fluid, and then the lymphatic vessels and taking that fluid, if there's like too much, is that when it comes out of the?
oh, I see what you're saying. Yeah. I mean, that could be one of the, definitely if you're getting more fluid buildup there coming into the area there, you can get a loss, like where you get the draining off you're talking about. Yeah. And sometimes too, it depends on if we were to kind of block this area off, because that's what your body's going to do. It's going to try to isolate this area here. And so when you start to move, sometimes you'll get, what pus is. Pus is predominantly cell debris and tissue debris, right? And partially also the pathogen there. And so that movement can push that material out of there because there is a pressure difference there. So yeah, we'll see that. But we're trying to just, one, wash kind of clean this area out, decrease the fluid so we can get, ideally, we want to get as much stuff into the lymphatic system as possible so we can expose it to the remaining cells in the immune system there in the secondary lymphatic structures. Yeah, go ahead. Interesting question. I can't remember if I have it on this slideshow or not. An abscess is usually kind of like what I was talking about. You'll get this walling off of this isolation there. Usually the fibroblasts will do that, and they'll create this kind of like collagen blockade. And then on the inside of the abscess, because usually, basically what an abscess is when we're trying to isolate stuff, usually infectious agents, and then the fallen soldiers of the immune system. So what we'll do is we'll start to put almost like a covering around it. And so that's what we'll see. We'll see this formation there around that area to isolate it off. And then the processes are still occurring inside of that area, right? But it's separated from the healthier tissue on the periphery. So usually an abscess, again, your body's trying to, it's like a contagion, What do we want to do? First thing you do, isolate it. And so that's when an abscess occurs. Yep, great question. All right, so let's go a little bit more clinical here and talk about, the different signs that you can see during inflammation. If you go through this, it all makes sense. Redness, right? Increased blood flow. Same thing happens when you exercise. You get vasodilation, more blood to the area, especially for facial flushing. Heat, all right? Heat I find interesting. Why? Increased blood flow. What's significant about that? Remember, blood is 1 degree higher than your body temperature. It's around 99 degrees, right? Compared to your normal 98, roughly, right? So it's usually 1 degree higher. So that's going to increase area, but also think about all the cells in that area. They're working hard, all right? It's like us exercising. This is the cells' version of exercising. They're going to increase their metabolism because they're going to be making proteins to help deal with this situation, right? But also they're just much more active. So we'll see increased metabolic activity, but also we'll find out this environment, all right, actually is beneficial towards the cells. They like a warmer environment. Some pathogens do not. I don't know if you've learned about that in micro. I know I keep looking over at you, but because some bacteria hate, all right, temperature changes, right? And so you'll see your body turns up a fever to help kill off some of those. Swelling, well, we just talked about swelling. Plasma proteins leave the blood, they get into the tissues, and that fluid loss from your blood winds up in the tissues, and we get increased fluid there. So that's swelling. Pain, because you have tons of pain receptors. You remember back in chapter 6 when you were learning about the skin, you had different types of receptors, Pacinian, Merkel's receptors, but there's free nerve endings. And those free nerve endings are everywhere. They're in the epidermis, they're in the dermis. And so those and nerve receptors are very sensitive structure. You could lay a hair across a neuron and be freaking out, right? So by design. So we have these nociceptors and these pain receptors that, again, because of that fluid there, is pressing on those receptors, and it causes, all right, those receptors to discharge. And so you can see you got compression from the fluid, chemical irritants, kinins, prostaglandins, bradykinin, all right, even those microbes there. All those foreign chemicals will irritate those receptors by design to let your body know there's something going on there. And then, of course, loss of function. And loss of function is more to an extreme case. You kind of learned about this back in chapter 6. When you have damage to an area, 2 things can happen. You can get, all right, regeneration, which means the tissue that was damaged gets replaced by tissue that's exactly the same, and so it doesn't lose function. Or you can get fibrosis, i.e. scar tissue formation, right? And so in extreme cases, you'll get a loss of function because you get fibrosis occurring there. And so we're no longer regenerating that tissue, we're replacing it, right? With something that's okay, it's not bad, it's just not as good as the original, right? So keep that in mind. So I mentioned to you that I like inflammation, it's good, but it's bad when it's chronic inflammation, right? And so it doesn't even have to be chronic inflammation in the fact that you see significant, all these cardinal signs there, right? You can have minor inflammation, and especially in what I deal with, and I'm not going to go off on a big tangent here, but when you get inflammation in tissues, specifically, I'll mention joints because that's my specialty, in the joints over a long period of time can cause tissue degeneration. When you get tissue degeneration, you get compromised function. So that's why chronic inflammation, we do not want that to occur. Why? Because it can have bad effects on us. All right, let's talk about fever. All right, so normal body temperature, 98.6 degrees. All right, and so here, when we're talking about a fever, normally we're referring to an increase in that body temperature there. What causes that? Pyrogens. Pyrogens are a name for a class of chemicals that your cells produce, right? Interleukin-1 is a common one, but there's several different types of chemicals, interleukin-6, all sorts. But interleukin-1 is one of the more famous pyrogens here that can come off of macrophages, gets into the bloodstream, goes to the hypothalamus and stimulates the hypothalamus, right, to increase your body temperature. How does it do that? Your hypothalamus releases prostaglandin E2, and then that causes your hypothalamus to change its set point. This is back chapter 1 stuff, all right? Your set point is the ideal number that your body wants to be. Like set point for blood pressure. What is that? Do you know the value? Yeah, 120/80. Yeah. Body temperature set point is 98.6, right? So what this will do is it'll cause it to change the set point and we'll elevate it. So we'll increase your body temperature here. And I'm going to tell you why, like what are the benefits for that in a second here. But our fever has 3 stages. So we're going to go through these right now, right? Obviously, fever onset, stadium, and then deferescence. Those are your 3 stages we'll talk about here. All right, let's talk about onset. So here's what happens. And this is why I tell folks it's okay to have a low-grade fever unless it's going on for weeks and weeks and weeks and months and months and months. That's not a good thing, all right? There's something causing that. But the first thing that we're going to see is vasoconstriction, right? And so we know in the skin that with vasoconstriction, it's going to decrease heat loss, right? Because we're going to conserve that blood and push it to the internal organs there. So we keep it away from the periphery. So we put it more towards the internal organs. And so that prevents heat loss because we want to keep your temperature elevated here. Because, and in some cases, I've had this happen before. If you ever gotten the flu, right? Has anyone here ever experienced the flu? Like the flu, straight up flu, all right? One of the worst things going on, but I had the chills. I had my fever elicited the shivering there. And again, what's the purpose of shivering? Generate heat. That's right. So that's the onset. So the stadium is we're going to maintain, all right, this high temperature. And again, certain bacteria, certain pathogens, I should say, like to operate within a certain temperature. So we make it, and hit in a hostile environment or an inhospitable environment, right? And so what we'll see here is this maintained temperature is going to help increase, right, our metabolism here. So we can continue to combat the pathogens, but also it helps us to get rid of, right, any harmful substances that may have resulted from the infection or are a result of the infection there. So our liver and spleen love to soak up zinc and iron because guess what? Your bacteria like to have those substances, and other pathogens want to use zinc and iron. That's why one of the things that they encourage folks to do, especially if you're near the 6th season, right, which we'll be moving into soon, is take zinc, right? Your body can utilize that, all right, to help with, all right, illness and whatnot. And then finally, the last event of fever is the deferescence. This is when we return back to normal. So the cells are no longer producing the pyrogens, which will then stimulate, all right, the hypothalamus to release the prostaglandins. So hypothalamus will not release the prostaglandin E2, and therefore the hypothalamus will start to cause a decrease in temperature. So it's going to want to get rid of all the heat. So it's going to stop the shivering. It's going to then cause vasodilation in the cutaneous blood vessels there. So blood flows to the surface of your skin and it gives off the heat there. And then sometimes in certain cases, you can get what they call cold sweats. And so that sweating mechanism there helps to cool you off, right, and cool you down there. All right, questions about that? This next slide you're going to love because you're going to see all the, so when anyone ever says to you, man, fevers are so bad, then you can just say this, boom. Here's why it's good, right? We slow down the reproduction of viruses and bacteria. We release our awesome interferons, alpha, beta, gamma, right? We're going to now stimulate, which we're going to talk to about here shortly, all right? Our cell-mediated immunity, which is part of our adaptive immunity. We're increasing the metabolism of a lot of these cells and these structures, so that's going to help with tissue repair. And we just learned about this stuff here. We are going to produce more cell adhesion molecules on the surfaces of our endothelial cells. And so this will help, all right, to, again, migrate our cells, but more so in our secondary lymphatic structures here. Go ahead. You look like you got a question. No? Okay, cool. So this is one of the reasons, well, not one of the reasons, this is several reasons why I don't have a problem with a low-grade temperature, okay? 100 degrees, 101, okay? I start to get a little bit suspect around 102. 104, I start to get concerned. 106, you better believe I'm concerned, right? 108, that's bad. I'm trying to think. The highest temperature I've ever had is 104. Has anyone had one higher than that? 105? You gonna one-up me? What a one-upper over here. All right, fair enough. All right, so let's talk about, all right, what are some of the bad things that can happen? Like I said, low-grade fevers, not bad. Okay, we get around 102, start to just be mindful, pay attention, see what's going on. 103 to 104, now you have to, you know, if it's happening in a kid, I definitely might give them a Tylenol. Okay, just to, I know it's not, but just to see if that affects the fever, right? If not, then I'm going to the doctor, right? Then obviously anything over 104, that's where we start to get worried. So when we're talking about high grade fevers, that's a problem. I mean, because one of the things that we talked about, we saw it early on in chapter 2, even though I know that a lot of you didn't have me as a professor. One of the things I'm always concerned about with your pH values in your blood is we got to worry about proteins. And when your pH gets really low, which means it's acidic, or even if it's really high, which becomes basic, you can get that denaturation of proteins. And basically, that's just the proteins start to unravel. And when that happens, they lose function. And when proteins lose function, we lose a lot of ability to do certain things here. So we have to be careful with denaturation of proteins, right? But also, when we talk about metabolic pathways, that's certain physiological processes that occur in your body that now we've disrupted because the fever is so high there. So we have to be concerned about when a fever gets high enough, seizures, loss of consciousness, and obviously the one that everybody knows is death, all right? It gets high enough there. So you could, what the main structure that we're worried about is the brain. That's the main organ there. So you get up to 106, you have to worry about seizures occurring. And then obviously things 108 or higher, you're cooked. Go ahead.
Like what's going on with your body? Like you got COVID, you like at 4.8, and from like 1 to 6 a.m. I had given him like every form of medicine for fever break.
Yeah.
You can think of, and at one point I was like literally putting like these ones, like he was like, like so much heat was coming off of me.
Yeah.
It woke me up, sleep aside, we had a fever. And it would not break from that time, like no matter how much I gave him. What was
Again, I mean, pretty much whatever the cause for that fever was, obviously it was the COVID, virus. And so how his body was fighting it, because his metabolism is skyrocketing because all of these cells are going to work on trying to combat that. And it's just, and his hypothalamus. And again, your body doesn't always get things right. You know, i.e. bone spurs. You ever seen bone spurs in people's feet or in their back? That's because the body grew too much bone in that area and it didn't need to. So in some cases, all right, individuals, their hypothalamus can get carried away, jack up the temperature, and just keep doing it as long as it deems that there's a threat there. So did you give them an ice bath or anything?
Like when I said I had every place that I've been,
Oh.
on every single part of his body.
Right.
It would not break.
Yeah.
Well, I've started laying hands on him and praying that way.
Yeah.
I'm
Pray it away, pray it away. Yeah,
yeah,
yeah. But what I tell folks is when all else fails, professional athletes do this all the time, not because of a fever, just fill up the bathtub with freezing cold water, get everything that is possibly ice related and dump it into that bathtub and stick them in that bathtub. I guarantee you that will help. All right. I'm not saying it will stop the fever, but it'll help them not get out of control. And that's one of the worst. I shouldn't say one of the worst, but it's one of the better ways to help manage an out of control fever like that. So next, if it ever happens, get him in the bathtub. But he'll hate you for it, but he'll love you for it afterwards. I'm telling you, he will. He will. All right. So this comes with what you were asking about abscesses, and I thought I had this in here. So this is, I'm going to skip down here to the bottom here. So actually, let me go through and talk about what pus is. And this is kind of what I was talking about. Again, when you have a battle, which is an infection, all right, you have a battle that goes on, there's going to be casualties. So those casualties include, right, the pathogens that were killed, the dead soldiers that your body had made to go and fight the war, and then the damaged tissue, right, that are part of the area where the battle was fought. So all that stuff is stuck in a certain location there. So a lot of the times, depending on how significant the whole infection was, a lot of that stuff gets cleared away. The lymphatic system will wash it away, it gets moved out of there, or your skin will eventually just resorb a lot of that and just kind of break it down. Now, in those cases, like what Willie brought up here, right? If we can't clear that away, we get the abscess, and then your fibroblasts come in there and they wall it off with collagen. Yeah. So this. Are you? Oh yeah, they're awful. Has anyone here ever watched the Dr. Pimple Popper? I think is the famous. Yeah, I've never watched it. I mean, that stuff. See, I know some people like that stuff. I'm not a fan, but yeah, that's what they're going to do. They're going to squeeze these things off. And again, the collagen, you compromise the collagen fibers, you can squeeze that material right out through wherever Dr. Pimple Popper is doing her thing. She'll evacuate the area. A lot of times you just go in and see a surgeon, they'll kind of cut it right out of there, right? But again, in some cases, in certain, does anyone here have heard of sarcoidosis? It's a respiratory infection, usually in the lungs. And what will happen is your macrophages will fight the infection, and then you have cells inside the lung that will literally encase those infections, all right, off into an abscess there. And a lot of times it's able to seal it off and it just leaves it alone. Problem is, you know, you have that pathogen that's still there. So if you were to ever rupture one of those nodules, you can release the pathogen back into the lungs. So that's always a consideration, but that's usually one of the situations that you'll see with sarcoidosis there, abscesses. Okay, so our first clinical view today is chronic inflammation. Chronic inflammation bad, acute inflammation not bad. All right, so again, chronic inflammation is going to be a period longer than 2 weeks. I've seen people a lot longer than that, right? But pretty much what you're going to see in acute infections, with acute inflammation, the predominant white blood cell is going to be the neutrophil. In acute infections and inflammation, normally those numbers start to drop off, and then at the tail end, or the longer inflammation occurs or an infection occurs, we'll start to see more lymphocytes. So neutrophils are acute, lymphocytes will be more chronic, but we'll always have macrophages, right? They're always on hand because they're the cleanup crew. So they're going to be doing their part for the immune response, but also they're going to be chewing up cell debris and kind of cleaning up the spot. Go ahead.
I was a dental assistant for a while, and I always wondered if the nerve of the tooth would die before it exists like a root. You get extracted, and then why would the abscess bring? I never
Why wouldn't it drain?
Where?
Oh, where would it drain? Well, yeah, there's lymphatics. I mean, the abscess will drain through the blood system there, the circulatory system, and there's lymphatics in your teeth. When we get into the digestive system, we'll actually go over the anatomy of the tooth, and you're going to see all the blood vessels that are there. So usually it's just too much for it to drain away, or you can't clear that material out of there because it might be too large to get into those vessels there to get washed away. So usually or you don't have ample drainage from that area there. Go ahead.
I'm like,
Dawn does a lot of amazing things, but I don't know about washing away an abscess. All right. I mean, you can clean up oil in the ocean with it, but you can't. But yeah, that one I don't know. I couldn't tell you. That's an interesting one. I mean, I could speculate. So we'll see a lot of chronic inflammation and overuse injuries. A lot of our athletes, for sure, all right, will deal with that. So especially, remember I was telling you, chronic inflammation is bad, and this is where we get that osteoarthritis, right? Which is eventually it's called OA for short, but that's arthritis, okay, in your joints. And so that is it's an inflammatory type of arthritis. You have inflammatory and non-inflammatory, but osteoarthritis is an inflammatory type of arthritis that occurs over time, right? Because chronic inflammation has occurred there. It's a condition. It's more of a condition. Yeah. Chronic inflammation is just, inflammation is sticking around, and I won't say we don't necessarily know why, but it's lingering over a period of time. And a lot of the components of that inflammation can be detrimental to certain tissues if those tissues are exposed to those chemicals over a long period of time. And it starts to cause wear and tear on the joints there, amongst some other things there. So when you have chronic inflammation, something is not resolving. We always look to the cause. Why is this not resolving? Is something happening because the pathogen is still there, or if it's not there? Hold on one second. So let's talk about our adaptive immunity. So depending on how much time I have here, time always goes by so fast. All right. So did you say 10 minutes? All right. That's what you're giving me? Gotcha. I will not disappoint. So, dang, this is the worst negotiation I've ever had. All right. So adaptive immunity, as you recall with adaptive immunity, all right, it is specific. So keep that in mind. We are going to be dealing with, all right, specific responses by a specific cell type. T lymphocytes and B lymphocytes. But here's the thing. What is it going to be responding to? This. I'm going to define what an antigen is in a little bit more detail, but that's what it is. An antigen is usually a foreign protein or a polysaccharide, sugar, right? But it's going to be something foreign that it will initiate a specific response from a specific cell. Innate, non-specific, right? Non-specific there. So the thing is, when we talk about the immune response, we're going to see those T and B lymphocytes and then everything that they make. So we're going to reintroduce perforins and granzymes, plasma cells, and antibiotics and antibodies. All right, so we're going to talk about all this stuff. The nice thing is, with our adaptive immunity, longer response. Okay, because we got to kind of create our army that's going to go in there and fight this battle. So it's like a recruitment type of scenario here. So you have two types. You've got cell-mediated, and so that's going to be our T lymphocytes. There's a test question in this. I can feel it. All right, so that's cell-mediated immunity. And then we have our humoral immunity, right? Think of the blood, right? And that's going to involve our B lymphocytes, which will turn into plasma cells. And those plasma cells will make antibodies. Yes, ma'am. Oh, I see what you're saying. Yeah. So and we'll get into that. I'm going to kind of answer that question probably in the next class. But usually what will happen is the first time that you're exposed to an antigen, it takes a longer period of time because your body's never seen it. So it's got to try to figure out what type of response. And then this is the beautiful thing I love about adaptive immunity is that you produce these cells, and we never heard about them really with COVID, but you produce what they call memory cells. And so these cells were made specifically for that antigen. So when you come in contact with it again, you already have kind of like a blueprint of how to handle that pathogen. So it doesn't take as long to do all the stuff that we're going to talk about here in this class. I'll show you. I'll talk about, yeah, it's actually really interesting. Oh, go ahead. Say that one more time.
It says antibody mediated immunity.
Or humoral. Yeah, yeah. That's an older term. Yep, that's an older term. So again, they're both the same thing because the antibodies are, again, just understand the antibodies come from the B lymphocytes, right? And I'll kind of explain it to you as we move through. That was your question too. Okay, all right. All right, so take a peek. Here's your adaptive immunity here, right? So you have your cell-mediated and the humoral, right? And so when we talk about cell-mediated, keep in mind what we're dealing with. T lymphocytes, humoral, or the antibody one that you're all describing, those are going to be the B lymphocytes here. And so we'll go through what's included in all this stuff. I have to tell you, the adaptive immunity is a little bit more complicated than the innate. So I'm going to kind of slow it down a little bit as we're moving through because we have a couple more classes to kind of go over this with you. All right, but just I'm going to do my best to take it nice and slow. And if it seems like I'm going real slow, there's a reason, right? And there's a reason because remember, I told you your third lecture test is one of the more challenging tests. I'm not going to say it's hard, but it's just one of the more challenging tests, right? So we're going to go through all this. So let's first describe, right, antigens. All right, so what is the actual definition of an antigen? Right, an antigen is going to be a substance that binds a T lymphocyte or an antibody. That's what an antigen is. But what does that mean? Like I said, it is going to be a protein or a sugar or a large polysaccharide. And so if you look at the examples up there, I mean, these all make sense. One of the things that they have, all right, in common is they're all foreign to your body. Right? So when we talk about the capsid, which is like that kind of envelope that goes around a virus, right? Virus isn't part of the body, that's foreign. So any protein that is found in the capsid there, the cell wall of any fungus or bacterial, which is made up of polysaccharides, right? We'll see that, all right, as a foreign substance. Bacterial toxin, all right, like Botox, for example, foreign protein, right? And then we'll see with tumors, though, yes, a lot of people say, well, a tumor is one of our own cells. Yeah, it's true, but it's an abnormal cell that's gotten out of control, and then it starts to express, right, abnormal, what we call cell markers or surface receptor proteins there. So point being is, it's not going to be the normal run-of-the-mill, okay, normal protein that is going to be expressed or sugar. So that's what an antigen is. So it's going to trigger, right, a T lymphocyte or an antibody. So a T lymphocyte, antibodies come from plasma cells, which were B lymphocytes. So both T lymphocytes and B lymphocytes are going to interact with antigens, so to speak, okay? A B lymphocyte will interact with it indirectly through the antibodies that they make, all right, as a plasma cell. All right, so this is very important, right? So specific antigens are going to be, are going to interact with specific lymphocytes here. Right, so this gets into kind of what I was talking about, self versus non-self, or foreign antigens versus self-antigens. All right, so again, we're going to go through a little bit of some definitions here just to kind of lay the basic groundwork here. So our foreign antigens, something that didn't come from our body. They're going to come, all right, the molecules that come from outside of our body are going to have a different sequence or different makeup of the proteins or the sugars that they express. Our DNA makes specific, all right, proteins for us, all right, whereas viruses and bacteria, they're going to express different, all right, proteins and whatnot. So self-antigens, you want to really be similar, basic, boil it down to the real easy understanding. Just go with the definition that's up here. Foreign antigens differ from the human body's molecules. Self-antigens, those are our own molecules. So that means with self-antigens, our immune cells will not bind to those antigens, which is good. We don't want that, right? Foreign antigens, our immune cells or our immune components will bind. So think of it like that, okay? Let's keep it real simple. So that means that our immune system has to be able to tell the difference between self and non-self, or foreign versus self-antigens. Right? And so autoimmune diseases are usually when the body can't differentiate self from foreign. So it just attacks our own molecules or our own cells there. All right. And if you've ever heard of Hashimoto's, we talked about that in 17. That's when your body makes your own immune cells will attack your thyroid gland. It'll actually go after your thyroid. And so, again, it varies. My mother has Hashimoto's. I tested positive for the Hashimoto's marker. And so I think my PA thinks that I have Hashimoto's. I'm refusing to give into that. But anyways, there's times when I go in and when they draw my blood to check my thyroid markers, that I do have an elevation in antibodies for Hashimoto's. So the point being is autoimmune disorders, your body will react to your self-antigen as it would if it was foreign. So when we're looking at an antigen, look at this little picture over here before you're writing anything down. Here's an antigen molecule. Here's that protein or that sugar, and you'll notice it's got these spikes coming off. Right? And so those little spikes coming off, right, those are called antigenic determinants, right, also known as epitopes. I like to use epitopes. And so these antigenic determinants are basically what your immune system is going to use, like when a natural killer cell comes along on its immune surveillance, or a T-lymphocyte comes along on its surveillance. It's going to quickly bind on to these things and see if it's self or non-self. If it's self, it releases it and it goes on its way. Then the immune cell moves on. If it's a foreign antigen, a non-self antigen, then it's going to stick around for a while. And we'll talk about what that process will be like, what's going to happen here, right? So just keep in mind that depending on what type of organism we're dealing with, they could have multiple, like this picture here shows several different types of antigenic determinants there, or epitopes. Okay, so you could have substances with one, or you can have substances with multiple. Okay, this brings me into lymphocytes. Lymphocytes. You know, to be talking about this for 3 hours, you guys are doing great, right? We only got about 20 more minutes. We're doing it. Just kidding. Okay, so let's talk about our lymphocytes. All right. Okay, so keep in mind, two types of lymphocytes that we're going to be discussing, the B-lymphocytes and the T-lymphocytes. And when we get into much more detail later on in this chapter, we're going to talk about the specific ways that B-lymphocytes will interact. But for right now, just understand that a B-lymphocyte is going to make direct contact with the antigen, right? So they're that frontline soldier that's out there actually fighting in a battle. Okay, so they're going to make that direct contact with the antigen, right? And so they can initiate the immune response that way. Now, our T lymphocytes, these are like the generals, all right? So they don't necessarily go directly into battle. What's going to happen is they're going to have the antigen brought to them. And guess who's going to bring it to them? Is it up there? No, it's not up there. Wait, what? You are now, I know. That would make me an antigen presenting cell, if that's the case. So antigen presenting cells, so the name APCs, they actually tell, all right, what their job is. Their job is to get, remember I told you about the dendritic cell? That's the cell that eats, all right, the infectious agent and then breaks it up and then sticks it all over its body, right? So those dendritic cells, right, will do that. And then they're going to look around for a T lymphocyte and they're going to show that T lymphocyte, hey, I've got all this stuff all over me, and the T lymphocyte will examine the antigen presenting cell. We'll get into that, right? So keep in mind that antigen is presented by another cell type. So it could be a dendritic cell, it can be a macrophage, yay, macrophage, and then it could also be, believe it or not, a B lymphocyte can play that role too. So they have 3 types of antigen presenting cells, okay? So we'll get into that. So one of the things that I will talk about, I'll just briefly mention it, make sure I got the time. All right, one of the things I will briefly mention is that our T lymphocytes have these things called co-receptors. All right, and so these co-receptors, like when you were in a, I'll quickly throw it, I'm full of analogies tonight. If you've ever been in a motorboat, okay, and you're going to be going to a dock to dock the boat, do you just tie the boat to the dock with one rope? No, because it'll be floating around and banging into stuff, right? So you're going to tie it to the dock. When you tie the boat to the dock, you don't use one rope because it's not stable. And so it could float around and bang into the dock or maybe bang into some other boats that are at the dock. So you're going to use at least 2 or more, all right, ropes to tie it to the dock, right? That's what these co-receptors do, right? They are going to help stabilize any type of interaction between the actual cell, the antigen presenting cell, and the actual T lymphocyte. And I'll promise you, I will get into more detail about that. Please believe me, all right? I'm just trying to throw some of this material out at you so you're not overwhelmed all at once. So when we look at the T lymphocytes, the 2 types that we're going to talk about are going to be listed up here. The helper T cells, I'm sorry, I apologize. I keep saying cells. All right, you can refer to T and B lymphocytes as T cells and B cells. So if I go back and forth between that nomenclature, understand it's the same thing. All right, T lymphocytes are the same thing as T cells. All right, so you have helper T lymphocytes, which are what we call, and you need to memorize this, CD4 cells. All right, just make sure that you know that. So helper T cells are CD4 cells. And the other type of cell, which I think is a cool name, cytotoxic, right? Cell killers, right? Cytotoxic T cells, they are also known as CD8 cells. And when we talk about CD4 and CD8 cells, we're referring to the type of co-receptors that I just mentioned a few moments ago. That's what we're talking about. And I'll show you this all here before we go, because I'm going to wrap it up here. All right, so just like the name says, the helper T lymphocytes literally assist or help, right, in both cell-mediated and humoral, all right, and even in innate immunity. So they play a role in all the different types of immunity in the body. Why? Well, they can activate 2 of the cells that we talked about that are heavily involved in innate immunity. So that's how they're able to cross over from adaptive and to innate. Because remember, we said that natural killer cells and macrophages are part of the innate immune system there, right? Remember we talked about the cell specific, not cell specific, but the cells that are specific, right, to certain responses. You have a question. You look like you're puzzled.
You said a cross-original fire to.
It dips its toes into both worlds, right? How does it do that? It can activate these 2 cells here. Remember we just talked about natural killer cells and macrophages and all the stuff that they do. Remember the natural killer cells release the granzymes and the perforins and punch holes into unwanted or unhealthy cells?
No, I'm fine.
Yeah. Okay.
In my head, I would feel like it would.
Yeah.
It's.
Okay, I know. I hear you. I hear you. No, I get it. All right, but just kind of keep that in mind. All right, the helper T. This is why, with conditions like HIV and AIDS, it takes out that class of cells, and that class of cells we need, and you'll appreciate it more at the end of this chapter. But this class of cells can help all the different aspects of your immune system. It plays a role in innate immunity. We're going to learn about how it does cell-mediated immunity here shortly, and then also how it plays a role with humoral or antibody immunity there when we're dealing with the B lymphocytes. So just keep that in mind, that they're important. That's why we don't want anything to happen to these cells. And that's why HIV sucks on so many different levels. All right. So cytotoxic T lymphocytes here, or the CD8 cells, these guys, it's like their name says, They come in and kill stuff. And that's literally what they do. They come in and destroy other cells. How? Like the natural killer cells. They release chemicals and they just punch holes in cells, and they'll even tell other cells to go and beat up these bad pathogens there. Okay. So I mentioned this before, right? Some of the other cells that we won't really cover too much, but I want to mention them, are our memory T cell. Well, I will talk more about memory T cells. Memory T cells are important because when we come in contact with that specific pathogen, we don't want to forget about it. You know, we don't want to start from scratch the next time we encounter the pathogen. So we're going to make these memory cells to memorize that pathogen. So when we come in contact with it again, we can kick its ass even faster and better. That's right. That's how we do it. All right. And then we have what's called our regulatory T cells. And the regulatory T cells are going to be, we got to shut this thing down. Right. It's like the police coming to a party that's gotten out of control or it's gone on too long. We got to shut it down. And that's what the regulatory T cells do. They kind of call or decrease the immune response. All right. And we'll talk more about that. Let me just show you this picture and we'll call it a night. All right. So T lymphocytes and B lymphocytes. This is what we're looking at here. So if we zoom in here, right, we have our two types. Here you can see our helper T cell on the left. So you can see all these things popping off of the cell itself. These are the receptors. And so there's only going to be two types of receptors that I want you to know for this class. You have what's called the TCR, which stands for T cell receptor. This is the receptor. When I was talking about I'm talking too fast. I got to slow myself down. When I was talking about immune surveillance, when these cells are on a walkabout and they're just inspecting cells to see if they're self or if they're foreign, the initial receptor that contacts another cell or an antigen is going to be the TCR receptor, the T cell receptor. And then, remember what I said, like our boat, we want to stabilize that connection. This other protein here, called the CD4 protein, because we're dealing with our helper T lymphocyte, this receptor here helps to stabilize that connection. Okay, so that's our second rope for our boat. On our cytotoxic T lymphocyte, again, we have a T cell receptor. So that's going to make the initial contact between the T cell and whatever it is that it's docking with. And then we have our CD8 protein, and that's going to help to stabilize. I'll go into more detail about it, but I just want you to know you have these two classes, right, of receptors here. And then finally, we got our B lymphocyte. And so the B lymphocyte has its B cell receptors, and they kind of look like antibodies here, right? But we'll see those on the periphery there. So I just want you to kind of understand what we're looking at here, all right? And we'll talk more about it in our next class on Tuesday. Do you all have any questions?